How many species of animals the scientists have identified? 1.3 million living species of animals. In summary, animals are multicellular, heterotrophic eukaryotes with tissues that develop from embryonic layers. What is the nutritional mode of animals? Heterotrophs, that ingest their food, use enzymes to digest their food within their body. What is the nutritional mode of plants? Autotrophic eukaryotes capable of generating molecules through photosynthesis. What is the nutritional mode of fungi? Fungi are heterotrophs that grow on or near their food and that feed by absorption, release enzymes to digest food outside their bodies. Describe animals' cell structure and specialization. Does plant and fungi are multicellular eukaryotes? Yes. Does plant and fungi have cell walls? Yes. Describe animal reproduction and development. Most animals reproduce sexually, with the diploid stage usually dominating the life cycle. After a sperm fertilizes an egg, the zygote undergoes rapid cell division called cleavage. Cleavage leads to formation of a multicellular, hollow blastula. The blastula undergoes gastrulation, forming a gastrulae with different layers of embryonic tissues. In haploid stage, sperm and egg are produced directly by meiotic division. Diploid cell. A cell containing two sets of chromosomes, each set inherited from each parent. Zygote. The diploid cell provided by the union of haploid gametes during fertilization, fertilized egg. It is one cell. Cleavage. The process of cytokinesis in animal cells, characterized by pinching of the plasma membrane, the succession of rapid cell divisions without significant growth during early embryonic development, that converts the zygote to a ball of cells. The cell doubles. Blastula. A hollow ball of cells that marks the end of the cleavage stage during early embryonic development. In animals, it surrounds the cavity called blastocele. Gastrulation. An embryonic stage in animal development encompassing the formation of 3 layers: ectoderm, endoderm, mesoderm. It determines fate of embryo, a process in which one end of the embryo folds inward, expands and eventually fills the blastocele, producing layers of embryonic tissue. Gastrulate. An embryonic stage in animal development encompassing the formation of 3 layers: ectoderm, mesoderm, endoderm. Archenteron. The pouch formed by gastrulation opens to the outside via the blastopore. The endoderm within the archenteron will become the tissue that lines the digestive tract. Two main characteristics of eukaryotic cells: nucleus, enclosed membrane organelles. What protein is found within nails and hair of animals? Keratin. Many animals have underscore stage. Larval. Larva is sexually immature and morphologically distinct from the adult. It eventually undergoes metamorphosis. Metamorphosis. A development transformation that turns an animal larva into either an adult or an adult-like stage that is not yet sexually mature. A juvenile resembles an adult but is not yet sexually mature. How is a mole form? It is formed when endoderm is stuck between ectoderm skin cells and just gets stuck there. Homeoboxes. The genes that control animal development are similar across a broad range of taxa. All animals have developed genes that regulate the expression of other genes, and many of these regulatory genes contain sets of DNA sequences called homeoboxes. Hox genes. Most animals share a unique homeobox containing family of genes, known as Hox genes, play important role of animal embryo development, controlling the expression of other genes that control the morphological characteristics of the embryo. Who of the animal kingdom lack Hox genes? What do they have? Sponges. They have other homeobox gene that influences shape. How does the Hox gene appeared? In the ancestor of more complex animals, the Hox gene family arose via the duplication of earlier homeobox genes. Over time, the Hox gene family underwent a series of duplications, yielding a versatile toolkit for regulating development. What is one aspect that the Hox gene controls of the morphological characteristics? Formation of anterior-posterior axis. Summarize the main stages of animal development. In most animals, the zygote undergoes cleavage, which leads to the formation of blastula. Next, in gastrulation, one end of the embryo folds inward, producing layers of embryonic tissue. As the cells of these layers differentiate, a wide variety of animals forms results. What family of control genes plays a major role? Despite the diversity of animals' forms, animal development is controlled by a similar set of HOX genes across a broad range of taxa. What does the molecular clock suggest about when the ancestor of animals' kingdom diverged? The ancestor of animals diverged from the ancestor of fungi about 1 billion years ago. Who are one of the most closely related living organisms of animals? Choanoflagellates. What did the scientists hypothesize with the evidence that choanoflagellates are the group of the most closely relative to animals? Researchers hypothesized that the common ancestor of living animals may have been a suspension feeder, similar to present-day choanoflagellates. What are the 3 evidences that suggest that the ancestor of the animals was a suspension feeder? What are the geologic eras? Neoproterozoic era, Paleozoic era, Mesozoic era, Cenozoic era. What time frame is the Neoproterozoic era? 1,542 million years ago. Describe the Neoproterozoic era in terms of evolution of animals. Macroscopic fossils were found 565 to 550 million years ago, which were part of an early group of soft-bodied multicellular eukaryotes, known as Ediacaran. Some are sponges, while others may be related to cnidarians. Microfossil is found in China that dates to 575 million-year-old. This exhibit the basic structural arrangement of the animal embryo. Still do not know if is animal or closely related to animal. This era was a time of increasing diversity. What is the time frame of the Paleozoic era? 542 to 251 million years ago. What are the periods of the Paleozoic era? Strata, Cambrian, Ordovician, Silurian, Devonian. Describe the Paleozoic era. The increase in the diversity of animal phyla during the Cambrian was accompanied by a decline in the diversity of ediacaran life forms. What are the hypotheses that explain these? Predators acquired adaptations, locomotion, that helped them catch prey, and prey acquired new defenses, protective shells. Thus natural selection declined some groups and rose others. Increase in atmospheric oxygen, that animals with higher metabolic rates and larger body sizes improved, and harmed other species. The origin of Hox genes and other genetic changes affected the regulation of developmental genes. This made the evolution of new body forms. What is the time frame of the Mesozoic era? 251–65.5 years ago. Describe the Mesozoic era. Animal phyla that appeared at the Paleozoic era began to spread to new habitats, first coral reef in oceans. Some reptiles returned to water, origin of wings and other flight equipment in pterosaurs and birds. Dinosaurs, first mammals appeared, tiny nocturnal insect eaters, flowering plants, angiosperm, and insect both had dramatic diversification. Late Mesozoic. What is the time frame of the Cenozoic era? 65.5 million years to present. Describe Cenozoic era. Mass extinction of terrestrial and aquatic animals, dinosaurs and marine reptiles, rise of large mammalian herbivores and predators. Global climate cooled gradually. Put the following milestone in animal evolution in chronological order from oldest to most recent. A. Origin of mammals. B. Earliest evidence of terrestrial arthropods. C. Ediacaran fauna. D. Extinction of large, non-flying dinosaurs. C. B. A. D. Zoologists sometimes categorize animals according to body plan, a set of morphological and developmental traits. Body plan. In animals, a set of morphological and developmental traits that are integrated into functional whole. The living animal. Our interest in the study of EVO-DEVO interface between evolution and development. Some of the evolutionary changes of body plans appear to be early history of animal life. Basic features of basic body plan. Symmetry, organization of body tissues, body cavity. Types of symmetry. Asymmetrical, radial symmetry, bilateral symmetry. Asymmetrical. Does not have symmetry. X. Sponges. Radial symmetry. Body is shaped like a pie or barrel, with no front and back, or left and right, can be divided into mirror image halves by any plane through its central axis. Sessile, attached to soil or other object. Her planktonic, weakly swimming. X. Sea anemones. Bilateral symmetry. Two-sided symmetry. The animal has two axes of orientation, front to back and top to bottom, have dorsal, top, ventral, bottom, anterior, front, and posterior, back, sides. X. Arthropods and mammalians have sensory equipment concentration at their anterior end, including a central nervous system, brain, in the head, called cephalization, move actively from place to place, have central nervous system. Cephalization. An evolutionary trend toward the concentration of sensory equipment at the anterior end of the body. In animals, true tissues are collections of specialized cells isolated from other tissues by membranous layers. During development, layers give rise to the tissues and organs of the animal embryo. Three germ layers. What organism does not have true tissue? Sponges. The embryo becomes layered through the process of gastrulation. What are the germ layers? Ectoderm, endoderm. Ectoderm is the germ layer covering the embryo's surface, gives rise to outer cover and central nervous system in some animals. Endoderm is the innermost germ layer and lines the developing digestive tube, called the archenteron, gives rise to the lining of the digestive tract and organs, liver and lungs. Diploblastic. Animals that only have ectoderm and endoderm. X. Cnidarians and comb jellies. All bilateral symmetrical animals have underscore a third germ layer called mesoderm. Mesoderm. Middle primary germ layer in a triploblastic animal embryo, develops into notochord, the lining of the coelom, muscles, skeletons, gonads, kidneys, and most of the circulatory system in species that have these structures, fills the space between endoderm and ectoderm. Triploblastic animals also have an intervening mesoderm layer. These include all bilaterians. These include flatworms, arthropods, vertebrates, and others. Most triploblastic animals possess a body cavity. Types of body cavities: coelomates, pseudocoelomate, acoelomate. Body cavity: fluid or air-filled space between digestive tract and body wall. Coelom: true body cavity, derived from mesoderm, not contiguous. Cells on the opposite sides do not connect, no air gap between them. Coelomates are animals that possess a true coelom. Pseudocoelomates: animals that possess pseudocoelom, which is a body cavity derived from the mesoderm and endoderm. Acoelomates: triploblastic animals that lack a body cavity. Functions of body cavity: its fluid cushions the suspended organs, helping to prevent internal injury. In soft body coelomates, the coelom contains non-compressible fluid that acts like a skeleton against which muscles can work, enables internal organs to grow and move independently of the outer body wall. Grades: group whose members share key biological features. It is not equivalent to a clade, a group that includes an ancestral species and all of its descendants. Developmental modes: protostome development, deuterostome development. How can you distinguish the two types of development modes? By differences in cleavages, coelom formation, and fate of blastopore. Protostome development: in animals, a developmental mode distinguished by the development of the mouth from the blastopore, often also characterized by spiral cleavage and by the body cavity forming when solid masses of mesoderm split. Deuterostome development: in animals, a developmental mode distinguished by the development of the anus from the blastopore, often also characterized by radial cleavage and by the body cavity forming as outpockets of mesoderm tissue. Spiral cleavage: type of embryonic development in protosomes in which the planes of cell division that transform the zygote into a ball of cells are diagonal to the vertical axis of the embryo. As a result, the cells of each tier sit in the grooves between cells of adjacent tiers. Determinate cleavage. A type of embryonic development in protozomes that rigidly casts the developmental fate of each embryonic cell very early. Radial cleavage. A type of embryonic development in deuterostomes in which the planes of the cell division that transform the zygote into a ball of cells are either parallel or perpendicular to the vertical axis of the embryo, thereby aligning tiers of cells one above the other. Indeterminate cleavage. A type of embryonic development in deuterostomes in which each cell produced by early cleavages divisions retains the capacity of develop into a complete embryo. During gastrulation, an embryo's developing digestive tube initially forms as called which becomes the blind pouch, archenteron, gut. As the archenteron forms in the protostome development, in Italy solid masses of mesoderm split and form the coelom. In deuterostome development, the mesoderm buds from the wall of the archenteron, and its cavity becomes coelom. Protostome and deuterostome developments differ in the fate of blastopore. The indentation that during gastrulation leads to the formation of the archenteron. After the archenteron develops, second opening forms at the opposite end of the gastrulae. In many species the and become the two openings of the digestive tube. The blastopore, the second opening, mouth and anus. In protostome, the mouth generally develops from the first opening, the blastopore. In deuterostome, the mouth is derived from the and the usually forms the anus. Secondary opening, blastopore. Distinguish the term grade and clade. Grade-level characteristics are those that multiple lineages share regardless of evolutionary history. Some grade-level characteristics may have evolved multiple times independently. Features that unite clades are derived characteristics that originated in a common ancestor and were passed on the various descendants. What is the goal of researchers that classify organisms? Reconstruct evolutionary history. The two types of view of systematics about animal phylogeny are _. based on morphological data, based on molecular data. What are the agreement points between these two points of view? A different view has emerged from molecular phylogenies based on ribosomal genes, Hox genes, and dozen of other protein-coding nuclear genes, mitochondrial genes. Ictiozoan. Member of a group of animal phyla identified as a clade by molecular evidence, many are molting animals, characteristics shared by nematodes, arthropods, and others, secrete external skeletons. Exoskeleton. As the animal grows it molts, squiring out of its old exoskeleton and secreting a larger one. Determined by molecular data, other members outside this clade shed their exoskeleton. Ecdysis. Process of shedding the exoskeleton. Clades. Eumetazoans, Bilateria, Deuterostomia, Ecdysozoans, Lophotrochozoan. Member of a group of animal phyla identified as a clade by molecular evidence, include organisms that have lophophores or trichophore larvae. Some members, such as ectoprocts, develop a structure called lophophore. Individuals in other phyla, including mollusks and annelids, go through distinctive development stage called trichophore larva. Before. In some lacophoans animals, including brachiopods, a crown of ciliated tentacles surrounds the mouth and functions in feeding. Trichophore larva. Distinctive larva stage observed in some Lophotrochozoans animals, including some annelids and mollusks. Describe the evidence that cnidarians share a more recent common ancestor with other animals than with sponges. Cnidarians possess true tissues while sponges do not. Also, unlike sponges, cnidarians exhibit body symmetry, though it is radical and not bilateral as in other animal phyla. How do the phylogenetic hypotheses presented in the 3 based on morphological data and the 3 based on molecular data differ in structuring the major branches within the clade Bilateria? The morphological base tree divides Bilateria into two major clades, Deuterostomia and Prostomia. The molecular base tree recognizes three major clades, Deuterostomia, Ecdysozoa, and Lophotrachozoa. The body cavity is underscore non-contiguous. The coelom is not an underscore gap. Air. The animals that have bilateral symmetry have underscore dorsal side, ventral side, the right and left side, interior and posterior ends, cephalization, posterior tail ends, cephalization. Bilateral animals often move underscore and have underscore a underscore system, also they are underscore actively. Muscles and nervous system. Central nervous system, highly vascular because they are very active. What phyla does not have a body cavity? Why? Porifera, they do not have true tissues. Radial symmetry species does not have underscore front, back, left, right. How does the planarians move? By using cilia on their dorsal side.